All right, as I was saying, we're going from simple to complex. So we use the energy from the electron being excited in that photosystem by which to do that. And we use a little electronic cell to actually do the reducing of the NADPH. All right, so enough about that electron, enough about NADPH. Let's go back to these protons that have been collecting inside the thylakoid. As those protons collect inside the thylakoid, as you said, you eventually get to a point where there's more of them inside than there is outside, and that's a disequilibrium. Remember what I said what living systems do? They let Mother Nature have her way, like diffusion. Things have to flow from high to low, okay? But they hijack the system, maybe? They use the system, they use Mother Nature's natural tendency for, water to flow downhill, like we would, in a water mill. They use that to their advantage. So, for example, since Mother Nature wants to correct this disequilibrium, she wants the protons to go back into the stroma. A few of them do leak through the thylakoid membrane, right? But most of them go through our buddy ATP synthase, which I introduced you to in respiration, right? And ATP synthase does that. It synthesizes ATP. Up above in the red, this whole process is known as that's the light part, phosphorylation. That's what's going on right there, right? When you stick a phosphate onto something, it's called phosphorylation. Don't worry about this right now. I'll get to it in a moment. We'll come back to this slide. Okay, so we got our two outputs. We got our source of electrons to be able to reduce, to build things with, and we have a source of energy. Just like anything, right? If you're going to build something, you need two things. You need the materials and you need the energy by which to do it. Next slide is a nice slide, again, to kind of help you hopefully not confuse these two together. In essence, what's going on the inner membrane of the mitochondria, what's going on the thylakoid membrane of the chloroplast is the same thing. Some sort of an electron transport chain, and the mitochondria happens to be 3 pumps and the chloroplast 1, is using the energy from energy-rich electrons, either from food on the left, sunlight on the right, to move protons across the membrane, to actually set up a disequilibrium. All of this fancy stuff, glycolysis, citric acid, all this stuff was intended to do nothing more than set up a disequilibrium of protons so that ATP synthase can be put in their way to take advantage of Mother Nature trying to, right the disequilibrium to drive this reaction. Same exact process, basically. Okay? All right, so we got our ATP. We got our NADPH. We're now out here in the stroma, right? All that previous stuff happened on a membrane. Remember, membranes are important. They are important. I'm sorry if it sounds like I said they aren't important. You can't make ATP until you get protons to flow through an ATP synthase. And you can't get protons to flow through an ATP synthase unless you put a barrier between them, so there's more in one than the other. Makes perfect sense, right? If you didn't have a membrane there, you would never be able to have more protons on one side because there wouldn't be anything for it to be on a side of, if that makes sense to you, right? Okay, so what we got here in the next step is we're going to take those that energy that we used and that imbalance that we created, and we're going to take those electrons that we've been gathering, this electron carrier's been gathering, and we're going to dump them into the Calvin cycle. And in the Calvin cycle, we're going to take carbon dioxide, a simple low-energy molecule, and create food, a much more complex, higher-energy molecule. So we're engaging in an anabolic reaction. You need two things for an anabolic reaction: materials, energy. Okay? Simple as that. And of course, as I mentioned, once that's being used, it's going to cycle back to the light reactions. Now let's take a look at the Calvin cycle. Sometimes it's also called the Calvin-Benson cycle, by the way. This might look a little familiar. The words are different, but this is a cycle of reactions, right? Just like the citric acid cycle in the mitochondria. And you remember the citric acid cycle, its job was to harvest a bunch of high-energy electrons, gather them, and then dump them into those chain of proton pumps to make that disequilibrium so we can make ATP. This is a little bit different purpose. Right? Same sort of idea, though. Mother Nature is very clever this way. The material that you start with comes from a modification of the material you end with. So it's like always going to be able to turn this cycle. All right, carbon dioxide from the air, Plants get carbon dioxide from the air. Rubisco is an enzyme. It's a shortened name. It's like maybe a nickname. Rubisco, really, its full name does end in ASC. But Rubisco is an enzyme that takes CO2 as a substrate, RUBP as a substrate, and turns it into this product. Right? Enzyme plus substrate, enzyme plus product. That 3-phosphoglycerate. Now, I'm not real concerned about you memorizing the names of these, but the phases are important. Phase 1 of the process is called carbon fixation. In chemistry, when we say something's been fixed, it means it's been incorporated into something. And Rubisco takes the carbon dioxide, sticks it into RUBP to make 3-phosphoglycerate. So it's capturing, fixing the carbon. See what I mean? Now, to turn 3-phosphoglycerate into this next stuff, you're going to need some energy. This is a more complex molecule. There we go. ATP. And that came from the light reactions. And of course, the used is going to go back to the light reactions to be regenerated. So we build it up a little bit of energy. We also need some materials. There they are. From the light to the light. So we're reducing this material, this 1,3-bisphosphoglycerate. And what we reduce it into is G3P. I talked about that in respiration a little bit, but G3P was one of the products during glycolysis. Right? So what G3P is going to end up doing, follow the arrow that kind of gets filtered off for just a second, it's eventually going to be turned into food. Right? So that's the process we've been trying to describe here with photosynthesis. This second phase is known as reduction because that's what's happening. So first we fix the carbon, then we reduce the material, and we filter a little off and turn it into sugar. Now, notice something interesting, though. This little number on the front means how many are made. Only one of those is output. The other 5 is left. And if you're thinking, well, the whole purpose here is to make food, why are we not using all 6 of them? Well, Mother Nature is more clever than that. Mother Nature says, I'll take the 5 that are left over, add some more energy from the light reactions. This goes back to the light reactions, and I'll do phase 3. I'll regenerate the material I need to keep the cycle going, right? So does this being evolved and adapted in such a way so that all of those G3P molecules, only one of them is going to become sugar. The others are going to be left behind to basically create the material that Rubisco needs to bind to with CO2 to make the stuff you start with. So it's very much like the citric acid cycle, right? Where you end up with what you need to begin. I just find that so interesting. All right, do a little math here. Let me erase this. Do a little math. For one cycle of reactions in the Calvin cycle, you're going to need 9 ATPs, right? You're going to need 6 there, 3 there, one cycle. And you're going to need 6 NADPHs. Well, really quickly, if you look at that, you can realize that you're going to run out of ATPs before you run out of NADPH. You're using them 1.5 times, 50% faster than you're using the NADPH. Now, if you were to run out of ATP, even if you had plenty of NADPH, it doesn't matter. You still can't make food. Now, the whole 6 and 9, that just happens to do with the chemistry. Living systems have to adapt and deal with that. So the adaptation I'm going to explain to you is going to help you understand how does a plant deal with the fact that it's using the ATP faster than NADPH? Or does it just get to that point where sometimes it runs out of ATP and it just sits there until it makes more? Now, that's not a good thing because that means the plant is no longer metabolically active. Now, this is the same picture you saw before. You know what? Let me go back to that one. Photophosphorylation, non-cyclic. The non-cyclic part refers to the direction or the path of the electron. It moves through the set of proteins left to right, from point